Okay, welcome, my dear friends. Welcome to today's session. So very soon you are going to attend CPGET 2024 online exam, that is CBT test, computer-based test. So in this regard, we would like to invite today's resource person, eminent personality, Dr. Manjula Madam Garu, Assistant Professor, Department of Botany, RBVRR Women's College Autonomous. Madam, please welcome and share your skin. Start your session. Please welcome.
Okay, thank you, sir. Sir, is it visible screen?
Yes, madam. Please click on slideshow.
Okay.
Yeah.
Sir,
Okay.
Okay. So good evening, all. So today's topic is nucleic acids. Okay. So nucleic acids, they were first isolated from the cellular nucleus, hence the name nucleic acid. So and they are isolated from the nucleus, hence the name nucleic acid. So these nucleic acids are macromolecules, that is large sized molecules and huge polymers with molecular mass of over 100 millions. Okay, so molecular weight is 100 millions and it is the larger molecule, that is macromolecules, and these are the polymers. So generally, introduction. So generally nucleic acids, these are the larger molecules and which are essential to the all known forms of the life. And here they generally they are called as a genetic material. Okay, hereditary material or genetic material. And here the two main classes of nucleic acids are present, that is the deoxyribonucleic acid and ribonucleic acid, that is the DNA and RNA. Two types of nucleic acids are present, that is DNA and RNA. And here based on the sugar molecules. If the sugar is the ribose, it is the ribose derivative, it is the RNA. Ribose nucleic acids and deoxyribonucleic acids. So RNA means ribose nucleic acids and DNA means deoxyribonucleic acids. If the sugar molecule is ribose sugar, it is the RNA. If the nucleic acid which contains the deoxyribose sugar, it is called as the DNA. Okay, so next one is the here two types. So first one is the DNA. So introduction about this DNA. So that is deoxyribonucleic acid. So DNA, it is a double-stranded molecule and that is twisted into helix. So DNA, they are the macromolecular structures and composed of regular repeating polymers formed from the nucleotides. So DNA is a complex molecule that contains the information which is necessary to build an organism. So main components of these nucleotides are nitrogenous bases, deoxyribose sugar, and phosphate group. So these are the main components of this nucleotide. Nitrogen base is present and deoxyribose sugar. So generally in the DNA, deoxyribose sugar is present, whereas in RNA, that is ribose sugar is present and phosphate group. So these are the main components. And nitrogen bases, the basic structure, that is pyrimidine and purine. Nitrogen bases are pyrimidine and purine. So again in pyrimidines, that is cytosine, thiamine and uracil. So three pyrimidines are present, mainly cytosine, thiamine and uracil. So whereas the main difference between the DNA and RNA is that another difference, that is the pyrimidine thiamine is present in the DNA, whereas the pyrimidine uracil is present in the RNA. In place of thiamine, uracil is present in the RNA. So three pyrimidines. So purines are similar to both DNA and RNA, that is the adenine and guanine. So sugar, that is deoxyribose sugar. So the sugars which are found in nucleic acids are pentose sugars. That is pentose sugar has 5 carbon atoms, and deoxyribose which is found in the DNA is a modified sugar which lacking 1 oxygen atom. Hence the name deoxy. So here the absence of oxygen, hence the name deoxyribose sugar. So this is the structure of the deoxyribose sugar. So next one is the phosphate group. So a phosphate group is just a phosphorus atom bound to 4 oxygen atoms. So here the phosphorus which is attached with the 4 oxygen atoms, but it has many important roles along with the sugar and bases, and it make up the nucleic acid. So next, so here this is a phosphate group. So it is bound with this sugar molecule. So here this is a sugar molecule which is bind with the base. So nucleoside. So here sugar and okay, 1 minute. Okay. So next one is the nomenclature of nucleic acid components, that is purines, adenine bases, that is the nitrogen bases, adenine and guanine, that is nucleoside is deoxyadenosine, and guanine deoxyguanosine, sin, SIN. Whereas here nucleotides, it is deoxyadenylate and deoxyguanylate. So pyrimidines, the cytosine base, cytosine, it is a deoxycytidine and deoxythymidine. So this is the nomenclature. So here the generally this purines, it ends with the sign, whereas the pyrimidines, it ends with dine or dine. Okay, DINE, dine, cytidine and thymidine. And deoxycytidylate and nucleotides are deoxycytidylate and deoxythymidylate. So this is a nomenclature. So next one is this is a primary structure of the DNA. So generally that is a 5 prime to 3 prime end, that is here each this nitrogen base which is attached with the phosphate group, phosphodiester linkage. So next one is a secondary structure of DNA. That is this model was proposed by Watson and Crick model. So secondary structure of DNA which was proposed by Watson and Crick model, and it is called as a double helix model. So the side of the ladder which are made up of alternating molecules of phosphate and deoxyribose. So the bases which make up the rings of the ladder are attached by a weak chemical bonds called as a hydrogen bond. So here these bonds, so two strands of this DNA, they are attached with the help of hydrogen bonds. These hydrogen bonds are weak hydrogen bonds, and the DNA double helix, it is a anti-parallel, which means one strand which starts with the 5 prime end of the one strand is paired with the another one with 3 prime end of its complementary strand. So that is 5 prime to 3 prime, one strand which contains 5 prime to 3 prime direction, and another one is a 3 prime to 5 prime direction. So the two hydrogen bonds connect to thymine to adenine, and 3 hydrogen bonds connect to the. So here the bonding also generally thymidine which pairs with the adenine with 2 hydrogen bonds, whereas the guanine which pairs with the cytosine with 3 hydrogen bonds. So the secondary structure of DNA, it is a double helix model. It was proposed by Watson and Crick. So next one is the types of DNA. So generally 3 types of DNA are observed, that is aDNA, bDNA and zDNA. So aDNA. So aDNA, it is one of the many possible double helical structure of DNA, and it is the most active along with the other forms. So helix has right-handed sense, and these are shorter, more compact helical structure. So it occurs only in the dehydrated samples of DNA. So generally aDNA which is occurs in dehydrated samples of DNA, such as those used in the crystallographic experiments. Next one is a bDNA. So it is helical sense, that is it is also right-handed, and base pair almost perpendicular to the helix axis, that is 3.4 angstroms apart, and one turn of the helix is 36. So here it is called, here these minor grooves and major grooves are observed in this double helix model. So this one is the major groove, and this one is a minor groove. And here the length between two nitrogen bases is 0.3 nanometers. And here the diameter is 2 nanometers. Between the two strands, the diameter is 2 nanometers. So here the minor groove is 12 angstroms in bDNA, and major groove is 22 angstroms. So one turn of the helix, it takes 36 angstroms unit. So next one is the zDNA. So zDNA is one of the many possible double helical structures of the DNA. So a helix has left-handed sense, and it is most active double helical structure. So zDNA was first discovered in 1979. So certain proteins bind very strongly to the zDNA. So these are the types of DNA. So based on the what you call turning and also the length. So here these are the different types. So normal DNA, it is the zDNA. Okay. So helix, here the property helix handedness is in aDNA, that is right-handed. In bDNA, it is also right-handed, and zDNA, left-handed. So base pairs per turn, 11 in aDNA and bDNA 10.4 and zDNA 12. So rise per base pair long axis is 0.23 nanometers, and here bDNA 0.34 nanometers, and in zDNA 0.38. So pitch, that is 2.46 nanometers, and here 3.4 nanometers, and zDNA 4.56 nanometers. So diameter is 2.55 nanometers in aDNA, and in bDNA 2.37 nanometers, and in zDNA 1.84 nanometers. So major groove is present, whereas in zDNA, that is major groove is absent. Minor groove is present in aDNA and bDNA, and it is deep cleft in zDNA. So functions of the DNA. So generally, the main function is, that is hereditary material. So scientists have since discovered that certain proteins bind very strongly to the zDNA, and suggesting that zDNA plays an important biological role in the protection against the viral diseases. So the main role of DNA in the cells is the long-term storage of the information. Okay, main function is storage of information. So here, DNA, it is a genetic material, and that carries information from generation to generation. So, but how we know that the DNA is a genetic material? So some scientists prove the DNA as a genetic material. So here we will explore some different experiments which carried out for as a genetic material. So what are the properties of the genetic material? So if any substance, if it has a genetic material, so the what are the properties it should follow? So for a molecule to act as a genetic material, it should have the following properties. So first one is capable of replication, that is form its own replica, and it should be structurally and chemically stable. The molecule should be structurally and chemically stable, then only it becomes a genetic material. So it must have the property to changes due to the mutation. So due to the so it should have the property to change during mutations and be expressed in the form of Mendelian characters. So these are the main properties of genetic material. So although DNA is a genetic material in most organisms, in some viruses and RNA, in some viruses, RNA is a genetic material. So generally, DNA is a genetic material in most of the organisms. But in some viruses, RNA is the genetic material. So according to studies, RNA was the first genetic material. But since RNA is unstable, DNA evolved from the RNA with a chemical modification, and it is more stable and carry more genetic information. So first they identified the RNA was a genetic material, but it is unstable. RNA is unstable. Hence later, so this DNA, it is evolved from the RNA. So here the some experiments which are conducted to explain the DNA as a genetic material. First one is a Frederick Griffith experiment. So Griffith performed his experiments in 1928, and he was working with the Streptococcus pneumoniae bacteria. He identified two strains of bacteria, that is R strain, that is rough colony, and S strain. So when cultured on the plate, so he identified two types of strains. So R strain and S strain. So smooth one and rough strain. So S strain bacteria have a polysaccharide coat which gives rise to smooth and shiny colony. So S strain is virulent and cause pneumonia. Okay, so whereas the R strain lacks a polysaccharide coat and it gives rough colony. So R strain is non-virulent. So if the S strain is present, then only it causes pneumonia. So whereas R strain, it is non-virulent. It will not cause disease. So he performed the following experiment with these bacteria and saw the different observations. So S strain, which injected into mice. So by taking the mice as a experiment, so here he taken the S strain and injected into mice. So mice develop pneumonia and it die. So when S is virulent strain, so when it is injected to the mice, it due to it develops a pneumonia and it died. So next R strain. So when the R strain is injected to the mice live. Okay. So next, he conducted another, that is heat killed S strain. So by taking the, he killed the S strain by using heat, by heat experiment. So he injected into mice live. So Griffith found that heating kills the bacteria. Okay. So next, heat killed S strain plus R strain. So injected into mice die. So Griffith transformation experiments. So first, rough strain, non-virulent. So mouse live, and smooth strain when injected to mice. So mice dies, and heat killed smooth strain when killed. Here mouse lives, and rough strain plus heat killed smooth strain. Okay, rough strain is non-virulent plus heat killed smooth strain. So here the mouse dies. So conclusion of this experiment is that he concluded that R strain had been transformed by the heat killed S strain. So this happened due to the transfer of transforming principles from the S strain to the R strain. So which made the R strain virulent. So although significant, his observation did not identify the biochemical nature of the transforming principle. So he concluded that due to the transforming principle from the S strain to the R strain, so here the mice will die. But he didn't identify the biochemical nature of this transforming principle. So next, every MacLeod and McCarthy experiment. So they determined the biochemical nature of the transforming principle and identified by Griffith. So these people purified DNA, RNA, and proteins from the heat-killed S strain and determined which macromolecule converted the R strain into the S strain. Okay, so they continued the experiment here. So in last experiment, Griffith experiment, they didn't identify the transforming principle. So these three people, every MacLeod and McCarthy, they confirmed this identified the transforming principle. So these people purified the DNA, RNA, and proteins from the heat-killed S strain and determined which macromolecule converted the R strain into the S strain. So here first, they treated the heat-killed S strain to the protease, RNase, and DNases to break down the proteins, RNA, and DNA respectively. So he first, these people first they purified the separated the DNA proteins, and they treated with the DNA for the breakdown of the DNA. Here they used the DNase, and for the breakdown of RNA, RNase protein, and for the proteins they used the protease. Okay. So observation. So he observed that treated with the DNases inhibit the transformation of the R strain, and proteases and RNase did not affect the transformation of the R strain to virulent form. So conclusion is that they not RNA or protein, but this theory was not accepted by the biologist. Next, another experiment which is conducted by the Hershey and Chase experiment. So in 1952, Hershey and Chase proved that DNA is a genetic material by his experiment. They proved that DNA is a genetic material. So they used bacteriophages to experiment as follows. First, bacteriophage is type of virus that infects. So bacteriophage means it is a type of virus that infect the bacteria. So here they followed this protocol. So some virus grown on medium which containing radioactive phosphorus, and some grow on the radioactive sulfur containing medium. So they are taken some virus and they are cultured in the medium containing the radioactive phosphorus and radioactive sulfur containing medium. So virus have radioactive DNA that grown on the phosphorus containing medium. So because DNA contains a phosphorus but protein does not. So in contrast, so virus contain the radioactive protein, so which grown on the sulfur containing medium. So because protein contains the sulfur, but DNA does not. So these radioactive phages were allowed to infect bacterial cell and transfer their radioactive DNA or protein. So after the injection, so this viral coat or particles were removed from the bacteria by the centrifugation process. So here this is overall experiment. So here the cell sulfur labeled protein capsule. Here the cell which containing a radioactive phosphorus and sulfur. So after centrifugation, no sulfur in the cell. So whereas here phosphor labeled DNA, that is infection, and blending and centrifugation. So after centrifugation, phosphorus in the cells. So when the centrifuge these cells, so here phosphorus is identified, radioactive phosphorus is identified. Okay, so observation. So he observed that bacteria were radioactive, which infected with the viruses containing the radioactive DNA. So while the bacteria were not radioactive, which infected with the viruses containing the radioactive. So first they taken the viruses, one containing the radioactive DNA and one containing the radioactive protein. Okay, so when they infected to the bacteria, so one bacteria contain the radioactive DNA and another one contain the radioactive protein. So conclusion, Hershey and Chase experiment concluded that DNA is a genetic material which transformed from the virus to the bacteria, but not protein. Protein is not transformed from the virus to the bacteria. So RNA and protein. So DNA is a genetic material for the majority of the organisms, but there are some viruses that use RNA as their genetic material, such as the SARS, influenza, hepatitis C, polio, and retroviruses like HIV/AIDS. So they all have either single or double-stranded RNA. So they use it to replicate RNA in their life cycle. So generally, DNA is a genetic material for the majority of the organism, but some viruses, RNA is a genetic material. Examples for this RNA as a genetic material in SARS, influenza virus, hepatitis C, polio, and retroviruses. Okay. So prions are the infectious agents that transmit their characteristics through the protein. So this is about the DNA as a genetic material.
Any doubt, my dear friends? Any doubt? Okay. Madam, thank you very much. Am I audible?
Yes, sir.
Okay, thank you very much. Clearly explained about the structure of DNA and discovery of the DNA, and also why DNA acted as a genetic material, why RNA is not acted as a genetic material. Okay. Now, Madam will going to explain about the another topic.